Thank you very much. Thank you. Thank you very much indeed. It's really great to be back. Nobody ever invites me back, so it's just fantastic. Thank you. And I'd like to thank Bonnie very much as well, and everyone for inviting me, because Bonnie's been just simply wonderful throughout this process. She's one of the nicest people you can ever wish to deal with in an organisational context with respect to conferences. She's really lovely. You're going to be really disappointed with this talk, actually. I'm sorry about that. And the reason you are is that I'm not going to say, this is what you've got to do. This is how you've got to make your lives better. This is how you've got to reduce your risk of breast cancer. What I want to try and do is to talk to you about the risks and how they fit in. And if any of you were at my last talk, I'm sort of continuing it, because I've been researching in this area now for quite a while. I'm sort of continuing the ideas into a very, very different area now. Now, one of the things I'll do as well, I'll talk a lot of chemistry, and I'm really sorry about that too, and I'll put lots of molecular structures up. Don't worry, don't leave, don't go to sleep. All I want you to look at is the shape of these molecules. I don't expect you to understand the complex chemistry of them, but I just want you to see some of the shapes of them. So let's just kick off then, just by talking about the sort of the background to all of this. And I want to talk about a man called Charles Darwin, whom you will all have heard of. And Darwin basically developed the science of evolution. He began the science of evolution. And out of that science of evolution, he talked about selection, and selection of the fittest in the context of birds, for example, with the right sort of beak to peck out the right sort of insects from the right sort of tree in the right sort of environment. And at the same time, people were beginning to think about how this could be inherited from one creature to another, from a father to its son, and from a mother to its daughter, or a mother to its son. So the sort of idea of inheritance was becoming endemic, as it were, during the time of Darwin. And there was another man working at the same time, and I'll show you a humorous picture of him for very good reason. And he's called Lamarck. And Lamarck and Darwin basically hated each other. And Lamarck was saying, it's nothing to do with that Darwin junk. It's obvious, isn't it? If you're a giraffe, for example, and you need to eat something for the top of a tree, you will develop a long neck. Darwin said, crap, it's nothing to do with it. He didn't actually, he was a vicar. He said, crap, it's nothing to do with that. What it's to do with is the genes that you've got that are being selected in if you need to eat at the top of a tree. The genes for long neck will be selected in. So what Lamarck was saying is your environment will determine how you are, and what Darwin was saying is no, no, no. Your genes will determine how you are. So this is the concept of nurture versus nature, and I want to talk about that in the context of breast cancer and just look at some of the aspects of nurture and nature. So let's have a look at that in a little tiny bit more detail, begin to break it down. So basically, and you know this, I know, nurture is basically your experiences and nature is your genetics. I'm going to talk very little about genetics. I'm going to talk much, much more about nurture. And by experiences, I don't mean how well qualified you are, and I'm glad you lost that thing of mine. I'm not talking about how qualified you are. I'm talking about your life experiences, the environment you're in, the food you eat, whether you drink alcohol, and all of those sort of things. The sort of thing the last speaker was alluding to as well. So before we can start talking about that, and I'm really pleased this thing goes around. I didn't know it did until I opened it this morning in my hotel room. Isn't Google amazing? You can download all these things totally illegally. And aren't they fantastic? And there's this bloke here trying to get me to sign a declaration saying I give the copyright to you lot. These people would kill me. I'm sure they took a long time to write that. So here's a strand of DNA, and I want to just look at this DNA a little bit and what happens to DNA. In the convention of DNA theory and DNA technology, we've got basically a whole series of sugars going around on the outside here, and between these sugars are a series of bases. Don't worry about all this if you don't know about DNA. But these bases determine the gene. So the sequence of bases determines a particular gene. And a particular gene determines what happens in a cell if that gene is expressed. So that's the basis behind DNA. So if I change some of those bases in your DNA, then that might lead to a change that might lead to a difference in that particular cell. So if I change the bases in the bit of DNA that controls how fast your cells divide, it might mean that your cells divide faster. And that might mean that I've actually changed the bases to make an oncogene. But that requires physical, chemical changing of those bases in a way that I'm not going to talk very much about today. So let's have a look at those risk factors in terms of cancer and in terms of that DNA. I'm going to return to the DNA in a few minutes' time, but I want you just to see the basic structure of that DNA. So the first aspect is genetics, which I'm just talking about. If you change the sequence of genes, change the sequence of bases, then you can change the gene. And an example of that's an ONC gene. But also, other risk factors like environmental chemicals might be involved in some of these changes. That's what I want to focus on. So some of the chemicals might be carcinogens, and these are chemicals that do change that sequence of bases. They modify those bases, they chemically react with them, they change them, they make them different, they make different genes. But on the other side, there are chemicals, which I've just called others, which I hate actually, but I can't think of a name for them, that might modify that DNA in some way, that it gets seen slightly differently by the cell, but it hasn't converted it into a different structure completely. You'll see what I mean in a minute. And one example of these sort of chemicals are the things I've been interested in for years, the xenoestrogens, or chemicals that mimic estrogen. And I'm not going to spend the whole of my talk on them, but I'm going to look at them quite a bit because they're getting more and more implicated in the etiology of breast cancer as we go along. So the genetics then is basically Charlie Darwin, and the environmental chemical bit is Lamarck. And it's quite interesting that Lamarck is coming much more into vogue now, and I love this. I'd love to have actually met the bloke, and I love this cartoon, which was from the Times in 1890-something, which shows Lamarck's head on a giraffe's body, which I think is a lovely illustration of what he was actually talking about. So let's return to that bit of DNA then. And it might be that these sequence of bases, it'd be bigger than this actually, but the sequence of bases in this bit of the DNA might in fact be an ONC gene, and it might be a modification in those bases in one person, it might be in you, it might be in you, it might be in you, it might be in me, that makes my cells divide in an uncontrollable way. That's the nature bit, that's there, it's in us, you can't change it, we inherited it from our parents, tough. But what you could do looking at carcinogens is modify that DNA. So here's a piece of DNA drawn in a slightly different way. And in the middle here, you can see something in purple. That's actually a chemical that's inserted into the DNA. And when it inserts into the DNA, the system that's reading that DNA and making it into a protein, which I'll talk about in a few minutes' time, sees that and thinks, what the hell's this? It shouldn't be there. I'll translate this in a different way. Or the body's defense mechanisms, DNA repair mechanisms come along and say, there's a bit of a bulge here, don't like this, I'll take all this out and I'll replace it with the correct information. And in trying to replace it with the correct information, it replaces it with the wrong information. And then that means that that gene has been changed. And that's the process of carcinogenesis. Bit complex, haven't got enough time to go into the great detail of that. So let's just look at that in a simplistic sense now. So if you look at that piece of DNA and it's got some modification in it, the chemical in there actually is a chemical that's present in cigarette smoke that causes cancer, just so you know. And that's how it works. It actually inserts into the DNA. And that genetics, which can actually be in the gene, can then be converted via an ONC gene into cell division, which then might lead to cancer. And I've put breast cancer here, so it's relevant to today. On the other hand, as I say, a chemical comes along and that can then initiate that process in the DNA, and then something causes promotion, causes that cell with that initiation to divide and to produce a clone of those cells, which then lead to the development of that tumour. And that's the conventional idea of initiation and promotion in cancer. And that's the sort of background that I want you to have when I begin to talk about some of the chemicals in the environment that might have those effects. We just heard quite a lot about estrogen. We heard the word estrogen all the time. I was sitting there very annoyed you didn't show the chemical structure. How dare you? I'm a chemist sitting there hearing the words and not seeing the structures. So here's the, you're probably relieved actually. Well, here it is. This is the chemical structure of estrogen, which you've all been dying to see, I can imagine. And don't worry about its shape, its structure, just look at its shape. And it's got this bit at the end here, which is really characteristic. It's called a phenolic ring with a hydroxyl group on it. And the other end, it's got a 5-membered ring with a hydroxyl group on it. And it's these two groups that are really important in binding to the estrogen receptor and switching on cells to make them female, basically. We can talk about that a bit more in a few minutes' time. Just as an aside, estrogen is actually synthesized in the cell from testosterone. So that proves that men came before women, which is obvious. Dangerous. You see, you thought you were dangerous. That was even more dangerous. But I'm not. I'm by the door. I can get out. So the ratio between estrogen and testosterone is really important in a cell. So let's look at some molecules that might look a little bit like estrogen. And I talked about some of these 3 years ago. And we were only just beginning to study them then. An awful lot of water has gone under the bridge since then, and we know an awful lot more about them. So here's a whole load of chemicals. I could put thousands. The United Nations, WHO, report that's just been produced. And in that report, they say there are at least 8,000 estrogenic compounds. And the fact that the WHO are producing reports on these things is showing that the importance of them is becoming recognized. When I last stood here, it wasn't. So it's quite interesting. We've moved on a very great way. So here's some of those chemicals. I don't want to talk about them in detail, but I do want you to see the similarities between them and how they fit in. Here's DDT, the pesticide, and you can see it's got this sort of chemically structure, looks a bit like that, and it's got something that if you're a chemist, you can say, well, it might look a bit like that. This is a compound called genistane, which is present in soy, and it's very similar in many ways. And here's a chemical called bisphenol A, which is a plasticizer, present in lots of plastics, fewer now than used to be because it's been banned, been removed from a lot of countries' plastic productions, but it's still present in plastics and can actually leach out into food. I can't go into the detail of that, unfortunately. There's a lot of story around that. I don't want to mislead you into thinking that it's a huge issue. It might not be in its own right. And here's a chemical that we actually discovered, in quotes, which is, as an estrogen, which is a detergent that was used. It's now banned. And you can see it's got the hydroxy group there and the hydroxy group there and that there. You can see they can be fairly similar. Now, with the jiggery-pokery of PowerPoint, which I'm beginning to master, and my students are very proud of me, and I'm even more proud of me, I can now do things like this. Wasn't that good? I'll just do that again. You ready? You don't know how proud I am of these things, because it's all very well for these youngsters, but us oldies, we take a long time to adapt to these things. And I'm only just getting there, I tell you. So you can see that when you superimpose these, there's a lot of similarity between them. And if I just go down each one individually, you'll see, as they begin to superimpose, that there's a very great similarity between those molecules. You might say, well, they don't look similar to me. But from the point of view of a hormone receptor, which sits around here, they've all got their hydroxyl groups in the same place. Some of them have got chlorines, but they're similar chemically. And they've all got this long bit of nothingness in the middle. And they've all got the hydroxyl at the other end. So they sit into that receptor very nicely and switch it on. So what do they do when they switch it on? Now, there's a conventional idea of what they do, and then there's the unconventional idea, which I'm also going to talk about. And I bet you, if I'm lucky enough to come back in another 3 years, the unconventional idea will then be conventional. Because this field is moving very, very fast indeed, and the evidence is building up very, very quickly indeed. So this is how estrogen works. Basically, the hormone, we're looking only at estrogen at the moment. This is steroid hormones generally, but we're looking just at E2, estrogen, estradiol. It's produced in several places, produced in the liver, it's produced in the ovary and in the testis in males. And it's excreted from those organs, gets into the bloodstream, gets to a cell. Bit of dispute about this bit, so I won't go into the detail. It then binds to this receptor in a lock and key fashion with those hydroxyl groups at one end going in, like the prongs on a key, and the long straight bit in the middle going in as well. And that receptor then goes along to the DNA, and it switches on genes on the DNA. Now you can begin to see then, if there was a gene on the DNA that was an ONC gene, and this estrogen switched it on, you wouldn't be in a very good situation with respect to the expression of that. Now the question is, where are these receptors? And those receptors are in many cells, but they're particularly in breast cells. Don't run away with the idea that's the only place they are, because it isn't. But that's where there's an awful lot of them, and we're focusing on the breast at the moment because of that particular tumour. Then when that gene is upregulated down here, it produces messenger RNA, which comes out of the cell, goes to the ribosome and makes proteins. And the proteins it makes are the proteins that were coded for by that gene. So there might be proteins that are responsible for feminization. There might be the proteins that manufacture estrogen. There might be the proteins used in pregnancy. Anything. That's what that's there for, to signal the production of those proteins. So that's all fairly obvious. But you can now get a bit more complex. I'm going to do this in great detail. No, I'm not. Don't worry. I looked at this the other day and I thought to myself, am I allowed to swear? I said, holy shit, I can't believe this. I mean, this is incredibly complex. And I just tried to pick my way around it, and it's almost impossible. But each of these little pathways emanates from the estrogen receptor, which is sitting here, being occupied. And don't worry about them, because I don't understand them. But each of these pathways involves changes in a way a gene is expressed. And the reason I show you this is just to show you it's incredibly complex. And there's an awful lot going on. When I last came here, I was suggesting it was just a gene that was switched on. And since then, a lot of this work has been done. But if you look at one of the particular endpoints of all these genes being cycled and switched on and off, is the cell cycle. And the cell cycle is that life-death cycle of a cell. Division, life, death. Division, life, death. And if you change that division, life, death cycle, which is what you're talking about as well, then you'll modify significantly the number of those particular cells. And that is thought to be the underlying principle of cancer. So a lot of those molecules I talked about will sit in that receptor and will cause this cascade of events which will lead to all sorts of things, but it will lead to cell cycling changes, and it will lead on this side to changes in cell death. Apoptosis is programmed cell death, so it'll modify those changes. The rest of it we don't really need to understand. Just go back a stage and just look at estrogens in the context of breast cancer. Now we're coming back to the conventional context. I was having a chat to a lady over coffee, and we were talking sort of about this, about estrogen receptors. So here's a breast cell, just shown very simply, nucleus in the middle, and there's the estrogen receptor on its surface. Not all breast cancers have estrogen receptors. And if estrogen occupies that receptor, it causes the cell to divide. Simple as that. So if there's an estrogen-positive cancer and there's estrogen around, it will cause that cell to divide. If you put something in to inhibit the binding of estrogen, like tamoxifen or raloxifene, they will stop estrogen binding, stop the cell dividing. So that's very, very simplistic, very easy concept to understand. Now we need to, just bring in all this information together, we need to look at why these estrogenic chemicals' effects are mainly in pre-puberty girls and postmenopausal women. That was also mentioned in the last talk. We're doing well here. We've never met before, but I'm following on quite well here. Let me just nip on to the next slide to show you that. Sorry, I'm one ahead of myself. If you look at estrogen levels through the menstrual cycle, you'll see, obviously, in women, and that was discussed earlier, these levels change drastically during that monthly cycle. And of course, not shown here, they change drastically during life and gently go up towards the end of the menstrual time. But if you look at the levels of estrogen in men, pre-puberty girls, and postmenopausal women, a little bit post, actually, not immediately post, the levels are around about 20 picograms per mil. So basically, women begin before puberty as hormonally rather like men, and they finish post-menopause as hormonally like men too. But if I gave you or anybody a dose of an estrogenic compound, say in your food, genistein in soy, perhaps bisphenol A from a tin of baked beans that have got bisphenol A plastic on the inside of the tin, then if you're a woman in this period of their lives here when they're having menstrual cycling, that woman, as you said earlier, is able to adapt to these changes in estrogen level. That's what she does. She's able to adapt to these huge swings. So a small amount of estrogenic compound will have no effect whatsoever. But pre-puberty, which we're not going to talk about today, and post-menopause, it's very different. So these estrogenic compounds then might well have an impact that you might not expect them to have in the context of femaleness in a cell. Now, I hope that makes some sort of sense. So now what we need to do is look at, A, the incidence of breast cancer. We all know that's just gently going up. And B, we want to look at how that might link to exposures and what those exposures might do in a gene context. So if we look at breast cancer, I showed this slide at the last meeting, which I'll put it up now actually, and it just shows this sort of gentle, if it is gentle, increase in breast cancer, particularly major increase between 1980 and around about 1993, I think it was. These are data for the USA, but most other countries follow that general trend. But if you look at that in more detail, around the world. That's quite interesting. And only found these data quite recently. They're only produced quite recently. If you look at the increase in incidence of breast cancer, it's going up all over the world, but in some countries, the increased incidence is very much steeper than other countries. But, and I couldn't find a graph to show this, I didn't have time to draw it from the raw data. If you look at countries like North America, like the graph I just showed you, and you look at the increase in incidence over a very long period of time, that country, as it showed on the last graph, did show an increase in incidence at this sort of steepness, but very, very much earlier. So there's obviously a change in this increase in incidence according to the countries that you're in. And if you look at those countries, did you notice that arrow came on? I'm very proud of that too. That required a whole 3-day course to do. In the old days, I had a blackboard and I just put an arrow on it, and it was all done. If you look at the degree of development of the countries, now this isn't absolute, but it's quite rough. The more developed countries are at the bottom here, the less developed countries are at the top here. The more developed countries got their breast cancer earlier than the less developed countries, which are actually doing the job now and getting their breast cancer up to the same level as that, or same incidence as that in the developed countries. That's quite an interesting piece of information. But the question is, what might be different between those countries? The developed countries went into the Industrial Revolution early. They produced a whole load of new chemicals, a whole load of pollutants, a whole load of different foods, modifications of foods, and all these things, which women in those developed countries have been eating and exposed to for a very long period of time. The developing countries are just starting to do that. And so I think this disparity between these two sets of countries is because of the exposure to chemicals in the environment and in food. So if you look at exposure to environmental chemicals, it probably is going up in these countries and probably quite static in these lower countries. Quite interesting. Don't know whether that's the case. Fits in with my theory, therefore I like it. So let's just explore that a tiny bit more and ask an important question. What effect might exposure to environmental chemicals have? I mean, that's what we're here for, to begin to just explore that. And there are certain questions we need to ask. Could those effects be multi-generational? Now, we've never talked about this before because this is new. I mean, people are only just beginning to see that your nurture, your Lamarckian effect, might be passed on to your children. Now, that's quite counterintuitive if you're a DNA biochemist, because the central dogma of DNA is you can't do anything unless you modify that DNA. I'll explain how that might happen. What about in utero? Could a child get exposed to these things in utero and modify its genes during that in utero process? I'm going to ask that question. I'm going to try and answer it, actually, and I think it's quite a definitive answer. And in order for an in utero effect to occur, do these chemicals go across the placental barrier? Because the developing child is very well protected by the placenta, and only very specific chemicals are allowed across. And estrogen is one of the chemicals that's highly controlled. And it has to be highly controlled because if it's a male child in there and it's getting a zap of estrogen from its mother, that's not going to help in its development of maleness. So I want to give you an example which is absolutely nothing to do with breast cancer whatsoever, but it's something from my research that illustrates some of the effects I want to then begin to map onto breast cancer. And this is related to exposures to chemicals. And if you're trying to look at exposures in humans, you can only look at effects relating to exposures a very long time ago. So I couldn't give you something now and then have a look at you and see if it's had any effect, because it probably wouldn't. It would probably take a very long time. So we studied a group of people that were New Zealanders that were in the Malayan emergency in the 1950s and 1960s. And the New Zealand Malayan Veterans Association have been absolutely fantastic over this, and they've given us quite a lot of money to study this. And they used a chemical called dibutyl phthalate. Again, just have that structural shape in mind for a few minutes. And why they used it, you can see them here, they're actually putting their hands in it, look, and they're smearing it onto their uniforms. And they smeared it onto their uniforms because they wanted to kill this little thing called a trombiculid mite. And this trombiculid mite actually carried bush typhus. And that's a pretty serious disease to get if you're out in the Malayan jungle and you've got a severe dose of very, very severe flu-like symptoms you get from it. So they used this chemical because this mite actually got in through the seams of their clothes. And so they actually smeared it with their hands on the seams and it killed the mite when it tried to push its way in through the seams. So these blokes were exposed to really very high concentrations of dibutyl phthalate. We did some work with them and actually looked at the incidence of a whole load of diseases. And we showed, quite interestingly, that in the war veterans' children, their sons, there was a greater incidence of a disease called cryptorchidism, which is undescended testes, and a greater incidence of a disorder called hypospadias, which is when the urethra goes out through the side of the penis instead of the end. These are developmental differences. And both of these differences are related to estrogen-testosterone ratios in the developing embryo. Now, I don't want to lose you in all of this because there's a very complicated biochemical story here, which I'm not going to go into. So I just want you to recognise that these diseases appear to be at higher incidence. And just to compare the incidence in the normal New Zealand population, you can see it's around about 1%. And in our populations, it was 5 and 2.5%. What I should tell you has been an awful lot of controversy about these results. I've received more hate mail over these results than I've ever received over anything, actually. And there have been more letters in the New Zealand Medical Journal saying what rubbish the results are than I think they've ever published on anything. But we've got to a stage now, I think, and there was a paper quite recently saying, OK, we accept that these changes have occurred, but the incidence is probably lower than that. But it's greater than the general population. Don't want to bore you with it, but it's to do with the sort of epidemiological calculations we carried out. The populations weren't totally matched, but it's impossible to get matched populations. So this is now accepted, which is why I show it you, but there is some controversy around it, and I need to be honest in telling you that. What we do know, though, is that if you expose a rat to dibutyl phthalate, a female rat to dibutyl phthalate, its pups have low levels of testosterone. That's interesting because that's showing mum's exposed, baby has effect. Similarly, interestingly, if you expose dad to DBP, pups have a lower level of testosterone. Interesting, because dad didn't give birth. Babies were never inside dad. So they couldn't have been exposed to that chemical. They must have had taken on the genes that have been modified by exposure to that chemical. Very interesting. So let me tell you what I think is going on. Now, we were talking before about cortisol, testosterone, and estrogen. This is the synthetic pathway. There will be an examination on this at lunchtime. So yeah, I don't know what you're laughing for. I say this to my students, and they all laugh too, and then there is. In fact, they had it on Friday. And there's a question on this in there. That's why I'm here. If you look at the way it's synthesized, the testosterone and estrogen are synthesized, they go down several pathways, but this is the main pathway. My little bulb's going, unfortunately, my little battery's going. The red line is the normal synthetic pathway for testosterone. And estrogen is synthesized from testosterone by a single enzymatic step. The glucocorticoid hormones, like cortisol, are synthesized in the same pathway, but via a different route. They all start off from evil, horrible cholesterol. We shouldn't like that, but if we didn't have it, we wouldn't have testosterone, wouldn't have estrogen, wouldn't have anything, basically. And there's this sort of division in the middle here. And what we think is happening is the enzyme at this division has been modified in some way by changing the gene to that enzyme, which is meaning that it's changing the flux through this pathway. So in the men that were exposed to dibutyl phthalate, their children are producing less testosterone because that gene that codes for that protein has been affected. Now the question is, how on earth can that happen? And that's where it gets sort of tingly spiny, because this is a really exciting, very up-to-date research, which I'm just trying to break into. But it's very difficult to break into this very complex area that's not my field directly. If we just extend that a little bit before we talk about what's actually happening there. In the embryo, what happens is a thing called a urogenital fold, which is the precursor stem cells, actually, a series of stem cells that are responsible for producing the gonads. And this is so simplistic, it's unbelievable, but it's sort of right, and it gives you an indication of what's going on. If that urogenital fold gets a whole load of androgens like testosterone, it makes the boy genitalia, and if it gets a whole load of estrogen, it makes the girl genitalia. So if you do something with the genes in between so that the androgen-estrogen level changes, it would modify the way that urogenital fold forms. And simplistically, that urogenital fold folds inwards to make the vagina in a woman in response to estrogen and outwards to make the penis in a male in response to testosterone. Again, very simplistic, but generally that's the sort of idea behind it. So if you think of the penis developing with the urethra at the middle, if the estrogen-androgen ratio is slightly wrong, then it makes that not develop quite properly. And that would lead to hyperspadias with the urethra coming out the side of the penis because it doesn't quite know which way to go. So that's the sort of general idea behind that. Now, you're probably wondering why on earth I'm talking about all this. And why I am is because it's giving us the basis to begin to understand how some of these chemicals might affect the development of breast cancer. And I'll give you some evidence for that in a few minutes' time. You don't need that arrow on there. Just because I could do it, I did it. And I'm very proud of that too. So just to reiterate the developing child stuff, it's the same sort of idea, but instead of the gene being modified in the dad and then passed on to the child, the gene is being modified in utero because of what the mother ate or the mother was exposed to. So if it happened to be a gene that was involved in breast cancer, it is possible you could switch that on in that developing child. And we need some evidence, first of all, that the sort of chemicals I'm talking about do actually cross the placental barrier, and indeed they do. And I had a wonderful PhD student working at the University of Auckland, in fact, where I was doing a collaborative project for quite a while. And his name was Biju Balakrishnan, and Biju, as you know, means small. He was about this big, and it was the most appropriate name. And he used to do a lot of work on human placentae, and he would take the placenta straight after parturition and put 2 cannula in so he could see where a chemical went in that placenta. And I just have a vision of this young bloke. He's a lovely guy. And he used to be, had to do it very quickly. And he would be tearing down the corridor in the medical school with a placenta covered with a big wet towel, keeping it warm to get it to the lab in time so he could actually do these experiments. And what he did was he put a cannula into the placental artery and a cannula into the placental vein, and then he perfused the placental artery and saw what came out in the placental vein. And that was a way of showing what would go from the mum to the child. It's not ideal because the placenta's dying at that point, but you can't take a placenta out earlier, so you can't do the ideal experiment, obviously. But he showed something really quite interesting. For bisphenol A, which is one of those chemicals that we're talking about having an estrogen-like structure, here's the levels on the maternal side, they're going down, and here's the levels on the baby's side going up, and the slope of the 2 graphs is the same. So that just means it's going straight across. So that proves that beautifully. On the other hand, genistein, which also looks like estrogen, it goes down on the maternal side, up in the child's side. It may not be obvious unless you do the calculations, but the rate that it goes down is greater than the rate at which it goes up. And that means that the placenta is taking it out, but not pumping it all into the baby. And it may well be that the placenta sees that more like estrogen, is controlling it, and doesn't see this one as much like estrogen. That doesn't matter. The point is that these things can get across into the developing fetus, and therefore can have an effect on their genes. And I had another student, Lisa Graham, who was working only on amniotic fluid in pregnant women, and she showed 67 different xenoestrogens in amniotic fluid, which proves that they're all going across the placental barrier. So this isn't supposed to be scaremongering and horrible. This is just as fact. You know, you eat your food, it ends up in your baby, basically. You are what your mother ate, as Peter Gluckman would say. So how can gene expression be changed by exposure to a chemical? Because now we've proved that there's always exposures, both of dad and of mum and of her baby. Now the question is, can these gene expressions, can these chemicals change gene expression that might lead to the development of breast cancer in later life? Now, if the ONC gene itself, if the gene itself for breast cancer is there, this is nothing to do with it. That's there, that's a risk factor. We're looking at other risk factors now that might add to that gene. So, as I mentioned earlier, this is just to refresh your memory. Here's some DNA, here's a chromosome unwinding to DNA. And this is a gene on the DNA, and the gene makes the protein, as I showed you earlier, and the protein has a function. And we want to look at how some part of that process can be changed. And the process by which we look at that is a system called epigenetics. And this is this stuff that really is exciting me quite a lot now. In fact, it's exciting anyone in anything to do with genetics and DNA at the moment. I am not an expert on this subject, but I'm trying to become one. And I'm having great fun learning this, I tell you. So let's have a look. We'll learn this together now. We'll have a look at what epigenetics is and what it actually does. So epigenetics is a way that you can modify the structure of a gene and modify the way that gene functions without changing the DNA basis. That's really very important indeed. So it can happen in several ways. It can happen by a process called DNA methylation, and I'll talk about that in a minute. And for example, a lot of methylation, hypermethylation, can affect the tumour suppression gene, which is a gene called TP53, and that then, if that tumour suppression gene is down-regulated, then tumours go up. And that's known. On the other hand, hypomethylation, reduction in methylation, can increase the expression of some oncogenes. These are pretty major statements, because what it's saying is, even if you have an oncogene, its expression could be changed by exposure to some chemicals. So when we're looking at risk factors, these aren't just adding together. One of them's synergistic. One's increasing the effect of the other. And there are two other things which I'll talk about briefly as well, and that's histone modification and nucleosomal remodeling. I love all these phrases. They're intended to confuse. So what I'll do is just show you how the whole thing works, and you'll see where these two things come in. If you look at the way DNA is packaged in a cell, it's in the nucleus, and at the time of cell division, it's all a great long strand of DNA, and the DNA is dividing into two, making new DNA. But when it's not in use, it's packaged, here's the DNA on the outside here, folded around a protein called a histone protein. And if you change the nature of that histone protein, you'll change the folding. And if you change the folding, you'll change the genes that are able to be expressed. Because again, very simplistically, to be expressed, the gene has got to be in a conformation that it can be opened and converted into a protein, basically. Don't want to go into the detail of that, but I mean, it's sort of obvious, isn't it? That if you mess around with the structure of that, you're going to mess around with the way the genes are expressed. So you need to understand that it's in that sort of format. And one of the changes, just to give you an example, just so you know we do actually understand fundamentally this, is that there's a particular amino acid called lysine, which has got an amino group for the chemists in the audience, if there are any, and that amino group can be trimethylated. And if that amino group is trimethylated, then it means that the bit of protein with that amino group on it can't interact with a bit of protein than it normally did interact with. So it sort of repels it. So if this histoprotein in the middle here gets those modifications, then it unfolds a bit. And if it unfolds a bit, it pushes the DNA out. And if it pushes the DNA out, it changes the way that DNA can be expressed and the genes are expressed. Is this making any sense? You're too polite, the lot of you are saying yes. You're like those dogs in the back of cars, you remember them? In fact, you're all asleep, really. Now, if you look at the surface of that structure, I love these. Aren't these fantastic? These are real pictures of what it really looks like. These are X-ray crystallographs that have been solved. So they're actually the protein with DNA wound around it. So the brightly coloured bits are the protein, and the grey bit you can see is DNA curled up around it. That illustrates even better that if you remodel that nucleosome by changing the protein, you would change the way the DNA is exposed. So all of these things are due to changes in the gene that don't involve modification of the DNA basis, but they do lead to changes in the way those genes are expressed. I want to just focus on methylation. And why I do is because there's a great deal of evidence that's shown that in breast cancer cells, there are very significant changes in the methylation of very specific genes, and that these methylations are almost certainly involved in increasing, or sometimes decreasing, but usually increasing the expression of those genes. So let's have a look at what actually happens in methylation. Again, there's a bit of chemistry going on here, but I hope we can sort of look at it in a simplistic way, not because I think you're simplistic, but because there's not enough time to go into the great detail. So here are the DNA bases all the way in the middle of the strand of DNA there. And I've got two lit up here. And those two bases that have been lit up have been modified, but in a very minor way. So they haven't been changed like a mutation where a great big chemical is attached to them. They've been changed slightly by methylation. And there's only one DNA base that can be methylated, and that's cytosine. And so you get a process of cytosine methylation. This is a cytosine molecule there. So if you imagine that cytosine molecule there is the one in this litty up bit here. And what happens is it gets an extra group here, look, with just one line there, which is a methyl group, added to it. Now you can imagine that if it does that, it looks quite different chemically. And it'd be recognized by the cell replication system quite different chemically. And what happens is you get a whole load of these methylations along the gene, and they're called methylation marks. And the bit that's getting quite exciting is that these methylation marks are like a binary code, on, off, on, off, on. And it seems to be that that's recognized. So it's that sort of code on the DNA code that's recognized as signaling switch on or switch off. So you're probably thinking now, why on earth is this bloke going on so much about this? You will see, I hope, eventually. So let's have a look what happens then if you cause those changes. Just think back to the New Zealand Malayan veterans, but also think back to the in utero exposures and all the things I've not talked about that might be happening if there are chemical exposures in the gut. So if, for example, the DNA in a sperm is methylated, is modified, or the DNA in an ovum is methylated or modified, or both are methylated or modified, that's unlikely, but it's a possibility. And these two come together during fertilization, then you'll produce a gene in the child that has got those modifications. But I don't want to tell you a fairy story side of this. In fact, the modifications change during that process. Do switch off if you don't hear this. And what happens is those methyl groups are hydroxylated. No one knows why this is the case, but they are. But it still shows the modification. So I've shown it now as a little star in there. So the offspring of those either one or both modified genes leads to a modified gene, which means that the expression of that gene will be different in that offspring to the expression of that gene in the parent. This is Lamarckian evolution that everybody thought was completely wrong until recently. And the environment has modified the gene there, and therefore the environment of that gene has been expressed in the child. Fascinating. Quite interesting, though, because if you think about it in an evolutionary sense, Darwin was sort of right, because what he would have said if he'd been here now, he'd be sitting there with his long beard, a bit grumpy, I think he probably was. Everyone reports him as being rather grumpy. He'd be saying, well, that's sensible, because if mum and dad have an experience, animals or humans, they want that experience to be passed on to their children because it's an important part of their child's biochemical learning process. So if you eat a particular food that might have a toxin in it or something, it would be good if your kids expressed a gene that allowed them to get rid of that toxin. So you can see why this might happen in an evolutionary sense. But the negative side of it, of course, is that it happens in a sense that we don't know what it might do. So, following on from that change in the DNA leads to an embryo developing, which also has a change in that DNA. And that embryo developing with the change in the DNA leads to a nice bouncing baby with DNA identical, absolutely identical to the parental DNA, but with modifications that mean that the genes might be switched on or switched off. They're all the same genes, but it's whether they're switched on or switched off that matters. So what we don't know with this little nipper is what genes are going to be switched on and switched off. And we don't know what that might lead to in terms of an outcome. It could lead to the really bizarre outcome. Like, this is our Prime Minister, believe it or not. This is a point, if ever I use this in my lectures at university, I'll go into this great long political discussion about governments and how awful they are and everything like that. But I won't do that now. But I thought it was quite a nice picture. I'm sure you see, that's copyrighted and illegal, I'm sure of it. So let's just have a look then at how chemicals that I'm interested in and I'm interested in telling you about might be fitting into this system. and what the evidence is. So remember I talked about, for example, bisphenol A. I'm just focusing in on two actually now, just to give you a specific example. And I told you it looked like estrogen, that's the black thing in the background, and bisphenol A is the red one in the front. And we've been through that, and you understand that, you know what it looks like. We know that that bisphenol A will actually sit in the estrogen receptor. And what I've got here is the binding cleft of the estrogen receptor, but not just an artistic impression of it. This is the real binding cleft. This is the estrogen receptor that's been isolated and crystallized in the presence of bisphenol A, and then X-ray crystallography carried out on it, and that's solved by a mega computer over in Australia in Melbourne. And you can see here's the bisphenol A molecule, and I'm not showing you the rest of it, but 17-beta estradiol, the female hormone, fits in exactly the same place. So it's showing it fits into that, and it's going to switch on that receptor. It's going to do all the things it does. That's one part of the story. That receptor then goes to the DNA, upregulates the genes, and it does that by methylating them. That protein that it binds to initiates the methylation. But also, it's possible that this bisphenol A in its own right might cause changes. So what we need to do is we need to look at our exposure to that chemical, and we need to look at animal experiments to show what else bisphenol A might do. And just to give you a very quick lowdown on exposure to this particular chemical, this is only one, millions out there, 8,000 we know of. It's used in manufacturing these sort of sandwichy plastic containery things, and it's used, as I said earlier, to line baked bean cans and pineapple cans and any old can, basically. And that's one of the really important human exposure routes. So we're all exposed to this and many other chemicals. And that can be shown quite nicely by looking at the levels in people. And these are Japanese people, quite interesting one, this. I was in Japan not too long ago, and I didn't realize that kids in Japanese universities drink, it's horrible actually, I hope there's somebody Japanese that loves this, it's cold coffee in a can, and they drink it like Coke. Quite intriguing. But the cans were all lined with bisphenol A. And if you look in 1992, you can see the levels of bisphenol A in these students' urine went up according to the number of cans of coffee they drank every day. So it shows that the amount you drink leads to an amount in your body, and therefore the effects that I'm talking about are related to the amount that you drink or the amount you're exposed to, the nurture side of you. In 1999, suddenly that flattened out. And the reason it flattened out is because the Japanese government legislated to stop the use of bisphenol A lining coffee cans for kids. Isn't that interesting? So legislation had a major effect. That's another thing I'm interested in, trying to influence governments to legislate, to change some of the exposures that are bad, basically. Now let's have a look at the animal side. So we've shown, I hope, that there's an implication in terms of exposure to chemicals that might be in the diet or the environment, and I've only used one, which is terrible. I should look at hundreds of them, but there are hundreds of them. But can exposure to these chemicals affect gene expression? Now, you know they can, because I wouldn't be telling you about it if I was just about to say they can't. So let's have a look at a particular strain of mouse called the agouti mouse. And there are a lot of other animals that are called agouti, and all it means is, I didn't know this until I looked it up the other day actually, the agouti means this sort of mottly, browny, brindledy sort of thing. Tabby cats are agouti gene positive, which is why they look tabby. They've got a combination of colours. And the reason for it is that the hairs are mainly black with a yellowy bit in them, or are mainly yellow with a blacky bit in them, just depending what sort of species it is. In the cats, it's mainly black with a yellow. In mice, it's the other way around. And the more expression, this is a bit simplistic, the more expression of that agouti gene, the more you get to this mottledy sort of look. So you get the sort of mottled look, not very good picture that, in that mouse. And you can see that there's a gradation in mice according to how that gene is expressed. So the gene is always there, but the amount of expression changes the colour of the mouse. Isn't this a great thing? Because that means we can look at the amount of expression according to chemical exposures by just looking at the colour of the mouse. Very good. I'd like to do some of these studies. These mice are very expensive. So right from the yellowy end, I'll be collecting on the way out, by the way. Yellowy end here, they're totally unexpressing, and at the browny end here, they're totally expressing. So what does all this mean? Interestingly, if you look at the methylation of the agouti gene and the genes related to it, I have to say that because it's not only one gene, it goes up according to the amount of expression. So that methylation is switching on that gene. Interesting, that's what I inferred earlier. Now, if you expose the females before pregnancy, not while they're pregnant, before pregnancy, and take them off the compound during pregnancy. So it's not a direct transfer across the placenta to this chemical bisphenol A, and I've selected this one. A lot of other chemicals have been looked at, but I've just selected this one to show you. That chemical causes a change in the agoutiness of those mice. And what it does is, as the dose goes up, the mice get browner and browner and browner. And as the dose goes up, methylation goes up. So what it's showing is that that chemical, bisphenol A, is actually influencing the methylation of a gene, which then influences the phenotype of the animal. Now, you're probably thinking, I came to hear about breast cancer today. This bloke hasn't mentioned it yet. But you can also see, in a breast cancer context, I'm going to give you some examples in a minute, that if there are genes involved, you could modify the expression of those genes according to the chemicals that you might get exposed to, which is why those chemical risk factors are rather more complex than just adding, they're synergizing. Does that make sense? You're still all nodding, this is great. The same happens if you give genistein, exactly the same. And these are all estrogen-like chemicals. They're all dietary constituents. Interestingly, genistein has been one of the good guys because postmenopausal women have found that eating soy sometimes ameliorates those awful withdrawal symptoms from estrogen that you have. You know, when you get that huge increase in estrogen, it goes down, all your receptors aren't occupied, and everything gets switched off, basically. And you get a withdrawal. It's just like a withdrawal from drugs, nothing, which is why you're grumpy and horrible all the time. But if you take a bit of genistein, that binds to that receptor, makes you feel better. But it also has this effect. So indeed, if it is involved in methylation of genes that are involved in breast cancer, it's a bad idea, which is why hormone replacement therapy is a bad idea, because it does just the same. It does exactly the same. Now that's gone white because I want to just turn my attention now to a study which has investigated this in breast cancer. And this is looking at a particular chemical called methoxychlor. And methoxychlor is one I haven't mentioned yet, but it's the only one that's actually been studied in breast cancer as far as I know. And here's methoxychlor. And if you look at the structure of that, you will, of course, all now be shouting out, gosh, it looks just like DDT. Well, it does, because it's actually a later pesticide that was based on the structure of DDT. And I told you that DDT has estrogenic properties, and methoxychlor has exactly the same sort of effect. And you can see there it is with estrogen, and there we are. I'm going to do that again because I'm very proud of it. And there we are. You can see the two have got those similarities of structure, which means that it will occupy the estrogen receptor site and do the other things that estrogenic compounds seem to do. Now, it's been quite an interesting study, which, if you can work this out, you're great, because I can't. But what it tells me something important, which is what I'm just going to talk about. Mothers, and this was in rats, exposed to, no, sorry, this is in humans, sorry, exposed to methoxychlor. Now, you might think, well, why on earth would a mother be exposed to methoxychlor? These were farmers or farmers' wives that were living on farms that were using methoxychlor several years ago, many years ago, in fact, some of them. And what the researchers have done, this is a fascinating study. It is huge and terribly difficult to understand. What they've done is looked at the expression of genes. And they've calculated the doses of methoxychlor that these people had from the blood levels, actually. You can do that quite easily. And they've shown that an increase in dose causes an increase in gene regulatory effect. Now, don't try and look at that and see them all going up and down in the same place, because they don't. Some genes are being switched on, some genes are being switched off. But what it's showing is this chemical methoxychlor, when mothers are exposed, is changing the expression of their genes. And all of these changed gene expressions are purely gene methylations. So it's changing it by changing the gene methylations. So what this is showing in one complex experiment, actually, but it's simple because it's more complex in the big picture of things, is that one chemical that we know we could be exposed to, that we would predict would have these sort of effects, is actually doing the things that we would expect it to do in genes to be having a gene regulatory change, perhaps in breast cancer. So let's just explore that just for a few seconds, a little tiny bit more, and then I'll begin to stop, which is what you're probably looking forward to. Here's chromosome 17, and this is the chromosome on which the tumour suppression gene is, and here it is. Isn't it interesting, actually, look at a chromosome and say, there's the gene. God knows how we know. I know how we know, actually, but it's quite a long, tortuous process working it out. That gene is called TP53, and it produces a protein called P53, and that protein interferes with the cell cycle. So under normal circumstances, P53 will be produced, and that will be slowing down the cycling of a cell. It will be making it not divide. And when the cell needs to divide, then that P53 concentration goes down, and that change is due to a change in the expression of that gene, because the gene is always there. And that change is due to a methylation change in that gene. So this gene is being methylated and unmethylated, methylated, all as a way of controlling the amount of P53, which controls the cell cycle, which controls the way the cell divides. So if you think of the methoxychlor experiment idea, the question is TP53 affected? And it is. And that was one of the genes, in fact, they showed, was hypermethylated. And if that happened, that would change the cell cycling. So it's only one example of what might happen in that circumstance. But it shows the sort of, I suppose, gradation from these exposures, these complex experiments that we're just beginning to do now, and the implications for breast cancer, which is what we're concerning ourselves with today. So in conclusion, then, we can say there's no question that environment can affect genes. And this is really only been discovered in the last 5 or 6 years. This is really very, very new stuff. And this is counterintuitive, but our intuition is changing very drastically indeed. And that the effect can be inherited. That's very new. But interestingly, and I didn't mention this at all, when you get inheritance due to methylation of a gene, rather than actual change of the gene, mutation, the inheritance only goes on for 3 or 4 generations. Isn't that interesting? So the agouti mice, for example, if you give them bisphenol A and they become more browny, you breed 2 of the browny ones together and they produce browny babies with 1 or 2 yellowy ones. And as you keep breeding the browny ones together, they'll produce more yellow babies. The methylation reverts back again. And if you think about that in an evolutionary context, it's obvious. Because if this is a big way of signalling from the parents to the children what it might expect to receive in its diet or its exposures in the future, they may not go on for more than a couple of generations. So it would be foolish to have that change go on forever and ever and ever. And it doesn't. Very interesting, I think that is. So what you're exposed to might affect your children. And that's a big thing we're all talking about at the moment. Good old Peter Gluckman, the chief scientist, never stops talking about it. You are what your mother ate. He coined that phrase, and he likes to tell you that every single time he speaks to you, and he's right. He did. And your exposure might be a breast cancer risk factor to your daughters. So we've got a situation now where you can blame your mum if you got breast cancer on 2 counts. You can blame her because you might have a gene, an actual gene for breast cancer, but you might have also inherited from her, following an environmental exposure, you might have inherited that gene switched on, as opposed to switched off. But your dad is not an innocent here. We never talk about dads, because they're on the sideline here, but they're not. Because what they're exposed to might also influence your gene expression, and might also influence the propensity for breast cancer. That's just my idea. That's certainly not been published anywhere yet. But I think that will be shown because it's certainly the case for everything else. And so I'll just leave you with that as a really rather sobering thought, in fact, that it's not only you, but your kids that you're affecting by your exposures. Thank you for listening. Thanks.
A couple of questions. about phytoestrogen,
Yeah.
which I think genistein is actually
Yeah.
phytoestrogen. Right? So
Yeah.
the effects of them are that xenoestrogens are far more long-acting in the body, as I believe, and have an amplified estrogenic effect, whereas the phytoestrogens have a short life in the body and have a much more mild estrogenic effect.
That's not necessarily the case, actually.
Not necessarily.
Phytoestrogens are xenoestrogens. Xenoestrogen just means a chemical that looks like estrogen that's not normal in the body. So phytoestrogens are from plants, whereas the other xenoestrogens, the synthetic ones, are man-made chemicals. And how long they last in the body depends on how quickly they're metabolized. And the phytoestrogens tend to be metabolized quite quickly by a process called glucuronidation and excretion. Most of the man-made ones tend to be slower, but that doesn't apply to bisphenol A, actually. It's incredibly quickly metabolized. And this is one of the arguments against not banning it. The regulatory authorities say it's metabolized so quickly that it goes away so quickly. So it's not as cut and dried as that. But in general terms, you're right. The phytoestrogens tend to have shorter-term effects because they got rid of more quickly. And the true xenoestrogens, we can call them that, tend to have longer-term effects. But their effects are very much the same.
So I did actually read a paper really recently, I think I've got it somewhere, about how, because well, I don't know if this is genistein or something else in the soy, if it was...
Could be cholesterol.
Young girls exposed early, it can actually prevent future breast cancer.
Yeah, it can.
By altering the
It can.
levels.
Yeah. I mean, I didn't talk about that at all, and I'll mention it quickly now because perhaps I should have. I quickly glossed over that pre-menstrual period and didn't talk about it. But one of my great interests is what happens actually during that time. And we know now by exposure to phytoestrogens and xenoestrogens at early age, you can actually lead to early puberty. And I'm not going to go into the details now, but there's a lot of link between the time at which a woman comes into puberty and their susceptibility to certain tumours in later life, like breast cancer, for example, and particularly uterine cancer. And I'm sure there's some connection between the two here. So to some extent, and I should really say these things, but I'm telling you a story, I'm biasing it, to some extent there's some positivity in some of these compounds, as well as negativity. But it does depend on the time of exposure. And since it's quite difficult to say, right, if you're exposed to between 5 and 10, you're okay. And if it's 10 to 75, you're okay. And then 75 onwards, you're not okay. We just don't know it to that definitional sense yet. So it's quite difficult to advise people about these sort of things. So I think it's better to leave it as a sort of confusing mess, to be honest with you. Because I'm very scared about coming to a meeting like this, any meeting, and saying, right, cut down on your xenoestrogens, and then we suddenly find out next week, in fact, it's probably rather a good idea that you have some xenoestrogens. So I'd rather people just understand it rather than me trying to preach what you should do. Did that sort of answer your question?
It did, yeah. I brought it up for that fact because just looking at the soy debate, people get so polarised about it.
Yeah.
And it probably is it seems that most of the research is towards it being beneficial, even for breast cancers and things, because it replaces the stronger effect that your estradiols and your other artificial xenoestrogens would have.
Yeah.
So I just thought, I just wonder how many more people like me sort of freaking out, thinking, please, can you do that? If you look
at the xenoestrogens, and I'll use the phytoestrogens as an example, and I'll use one non-phytoestrogen. The way they bind to the estrogen receptor is what's important here, and their estrogenicity, i.e. how many molecules of xenoestrogen it takes to represent one molecule of estrogen, what's important. And if you look at something like genistein, that's 10 to the minus 5 times the activity of estrogen itself. DDT is 10 to the minus 6 times the activity of estrogen. This is what you mean by milder. They have a much lower effect. So you need much bigger doses to have the same effect as estrogen. But I mean, estrogen in the body is working at nanomolar, 10 to the minus 9 molar concentrations. It's very, very low. And it's very easy to reach 10 times those concentrations with something like DDT. But you're right to point out that there's more to it than negativity. And just one final thing on the positive side, because I feel terribly guilty now. On the positive side is osteoporosis, classic. That is reduced by estrogen levels, and it only occurs postmenopausally as the estrogen levels go down. And if you replace some of the estrogen activity with something like genistein or cholesterol, it's thought to, and it's quite good evidence, to reduce the incidence of osteoporosis. So, it's another positive side as well.
Thank you.
You pays your money, you takes your choice.
One more question.
A man, oh my God. Can I shake your hand? They're terrifying, all these ladies out there. There's another one up there. I saw him. Yeah, there's another man up there. Hey, hello.
Thank you very much for your talk. There's some work done by Cavalieri looking at mutagenicity of the estrone metabolites,
yep.
and that is another mechanism for potential impact, particularly xenoestrogens,
yep.
And also the problems with lack of certain substances in nutrition, such
yep.
as iodine, which contribute to estrogen being metabolized exactly in the pathways. I'm just wondering any comments on that, please.
Yeah, I mean, I didn't go into that at all. You did a little bit. You talked about the metabolism of E2, and it's metabolized to a series of other estrogenic compounds, or estrogens, and one of them is estrone. And that just has that, I showed you the single bond O on the ring, it goes to a double bond O. So it's a tiny, tiny change, but a huge change in activity. I mean, it's many thousands, I think I'm right, times less active in terms of an estrogen, but there are questions about its gene regulatory effects. And, you know, looking at it, it could well be a feedback mechanism for stopping the production of estrogen by actually feeding back into an earlier gene and switching it off. Because the estrone is signaling, probably, that the estrogen is in high concentration, therefore is being metabolized. Now, I don't know a lot about this, not my field, but I know there have been quite a few papers recently looking at estrone levels. So I think you're quite right to mention that. It's this big balance between the whole lot. But when you talk about T4, and I keep saying you did this as well, you talked about T4, quite fascinating, because when you look at the molecular structure of T4, it's an estrogen mimic. And that, I've just written a paper on this actually, in a veterinary journal, believe it or not. But it sort of intrigues me. And there are some chemicals in food. One of them is a red dye that's used in maraschino cherries, actually, and also in cherry aids, which mimics T4 and changes the metabolic rate. It also mimics estrogen, but to a very much lower extent. And it increases the tumour load in rats in the thyroid. Now, I'm not trying to make a connection between these things, actually, but I just sort of lose the link. But there's something big there, because these things are clearly, as you said, they're clearly intimately involved in the way they behave in the body, T4 and estrogen. And it's interesting that some of the environmental chemicals have dual mimicry, and in fact, those two hormones do as well. That didn't answer any question, I know, but it's a link between the two.
Thank you. The point really I wanted to make was that there are potentially other reasons for avoiding xenoestrogens than the mechanism of methylation.
Yep.
And I think my sense is that given the epidemiological evidence for the association of high levels of xenoestrogens and rates of breast cancer, that I think we should be cautious, particularly with the non-phytone estrogens.
Yeah, no, I agree. Yeah, agree. I'm done. Thank you. Thank you. Thank you very much. Thanks.